A very good morning to Dr. Hafiz and everyone. My name is Kodiro. Today I'm going to present my presentation topic, which is a biological response towards toxic materials in food. First of all, I will go through the toxic materials in food. The toxic materials in food can be comprised of natural sources of toxicants in food and contaminants, which is listed inside the slide below. Next, I will present about the effects of toxic to our body. The effect of toxic can be divided into three categories, which is selective chemical effects, non-specific chemical effects, and special toxic effects. In selective chemical effects, chemical substances show their severe toxic effects under their lethal dose at definite concentration in a region of the body specific for them. While the target point is the place where the normal cell components or cell membranes where the chemical substances show their effects in the living organism. For non-specific chemical effects, some chemical substances do not show their toxic effects at specific organs selectively. However, they have a widespread effect. For example, strong acids and bases destroy all living cells by denaturing and precipitating the proteins in the cell membrane, while organic solvents are harmful to lipid membranes, causing dissociation of nucleoprotein complexes. For the special toxic effect, it can be divided into mutagenesis, carcinogenesis, and teratogenesis. In the mutagenesis, it is the alteration of DNA structure, which can be divided into destructive addition and substitution of DNA. Next, I will present about the toxic responses in our body system and organs. First, let us discuss about the toxic responses of the blood. The effect of chemicals on blood cells. The first effect is bone marrow suppression, which is the deficiency of all or some cellular elements in peripheral blood. This might resulting in decrease in production of cells or an inability of bone marrow to manufacture adequate numbers of these cells. Examples of the toxic are arsine, benzene, and sulfonamide. Second, the hemolytic anemia, which is caused by an increased rate of red blood cell destruction. This response may be chemically or induced by immune response. Examples of toxics are arsine, benzene, lead, methyl chloride, phenylhydrazine. The third one is the polycythemia, which is the presence of an abnormally high red blood cell count and increased hemoglobin concentration or increased hematocrit. Example of toxic is cobalt. Next, we will move on to the effects on hemoglobin. First is the methemoglobin, which is the iron in the kin of the hemoglobin is normally in the ferrous state of iron 2+, but it can be oxidized by certain chemicals to the ferric state, which is iron 3+. The resulting pigment is known as methemoglobin. Examples of causative toxic are nitrates, nitrobenzene, sulfonamide, and hydroquinone. Finally, the cyanide poisoning. Cyanide can inhibit the cytochrome oxidase, which interfere the electron transport chain, resulted in no formation of adenosine triphosphate. Examples of food containing cyanide are almonds, tapioca, millet, sprout, lima beans, soy, and spinach. Next, we will move on to the toxic responses of the liver. There are 6 types of hepatotoxicity. The first one is the fatty liver, which is also known as steatosis. It is caused by the excess accumulation of lipid in the parenchyma tissues. Examples of toxic compounds that cause steatosis are ethanol, carbon tetrachloride, yellow phosphorus, valproic acid, and bromobenzene. The second type of hepatotoxicity is hepatic death, which is the end result of excessive or prolonged exposure to hepatotoxic agent. The hepatotoxic agents that cause hepatic death are carbon tetrachloride, bromobenzene, microcystin, ethanol, trinitrotoluene, and trichloroethylene. The third type of hepatotoxicity is liver cirrhosis, which is the result of chronic toxic injury or inflammation of the liver parenchyma in which the damaged tissue is replaced with fibrous scar tissues. Examples of toxic compounds that cause cirrhosis are ethanol and methotrexate. The fourth type of hepatotoxicity is the hepatitis, which is the inflammation of liver occur in response to chemical-induced hepatic cell injury or as a result of viral infection. Examples of chemical compounds that cause hepatitis are isoniazid and nitrofurantoin. The fifth type of hepatotoxicity is the cholestasis, which is the reduction of bile formation or impaired secretion of specific bile components. Examples are dichloroethylene, methylene dianiline, manganese, organic arsenic estrogens, and ethanol. The final types of hepatotoxicity is the liver cancer, which is caused by aflatoxin, arsenic, and vinyl chloride. Next, we will move on to the toxic responses of the kidney. So we will discuss about the effects of nephrotoxin. Nephrotoxin could act on the vascular elements supplying a nephron or any portion of the nephron directly. The effects include transitory alteration in cell transport activity, such as glucoseuria and amino acid urea. Second is the secretory failure, and third is the failure to concentrate urine, which condition known as polyuria. Fourth is the cell death. Five is the necrosis, and finally renal failure. A nephrotoxin could result in vasoconstriction and ischemia, with decreased glomerular filtration and tubular transport. Nephrotoxin also can affect the glomerulus directly, with resultant filtration dysfunction, either increase or decrease. And finally, nephrotoxin could affect the tubular, proximal, distal loop of Henle or collecting duct activities, with resultant of resorption, barrier, or secretory dysfunction. Next, we will move on to discuss the toxic responses of the nervous system. So, a neurotoxic substance is any substance that capable of producing an adverse effect on the structural or functional components of the nervous system. The nervous system is particularly vulnerable to toxic insult since nervous tissue has a limited or negligible capacity for regeneration. Neurotoxins effect may be immediate or delayed, or permanent or reversible. Neurotoxins may elicit their effects at the molecular, where the neurotoxin may interfere with neurotransmitter synthesis, leading to reduced production and subsequent neuronal dysfunction, or cellular level, which alter the ionic flow across the nerve cell membrane, leading to inhibition of impulse formation and transmission. So, the neurotoxication effects including the motor effects such as convulsion, tremors, lack of coordination, paralysis, and weakness. Second one is the sensory effects such as equilibrium changes and visual, tactical, and auditory disorder. The third effects is cognitive effects such as confusion, memory deficit, and speech and learning impairments. The fourth effect is the mood and personality effects such as depression, excitability, irritability, restlessness, delirium, and hallucinations. The final effect is generalized effects such as loss of appetite, fatigue, and overall depression of neuronal activity. Next, we will briefly go through the toxic responses of the cardiovascular system. The example of toxic contaminants in foods includes the inorganic and organic one. For organic, arsenic may cause coronary artery disease, while cadmium causes hypertension, cobalt causes cardiomyopathy and pericardial effusion. Lead will cause hypertension. Mercury also, methyl mercury can cause myofibrillar degeneration. And finally, phosphorus can cause myocardial fatty change. On the other hand, organic chemicals such as alcohol may cause cardiomyopathy or fatty change. Nitroglycerin will cause coronary arterial spasm, and carbon disulfide will cause coronary artery disease, and finally carbon monoxide will cause myocardial ischemia. Last but not least, we'll go through the toxic responses of the immune system. The effects of toxins on the immune system including hypersensitivity, immunosuppression, autoimmunity, and neoplastic changes. Hypersensitivity can be divided into 4 types. For type 1, it is an anaphylactic reaction, while for type 2, it is a cytolytic reaction, and type 3 is an immune complex reaction, and finally the type 4 is a delayed type hypersensitivity reaction mediated by T cells. For immunosuppression, there are several medicinal as well as drugs of abuse and environmental pollutants have been found to have an immunosuppressive effect. For example, steroids. Steroids will cause lymphopenia with suppression of both T and B lymphocytes, as well as neutropenia, followed by administration of steroids. Steroids also suppress the phagocytosis and prevent the release of liposomal enzymes from the neutrophils. For the drugs of abuse includes alcohol, cocaine, cannabis, and heroin. For autoimmunity, it can be caused by antibiotics, which the penicillin can cause an autoimmune hemolytic anemia, which disappears upon cessation of therapy. Methicillin produces an autoimmune induced acute interstitial nephritis with circulating antibodies to tubular epithelium. In the hemolytic anemia, penicillin acts as a heparin. Last is the neoplastic changes. It is caused by aromatic hydrocarbons, which are associated with sarcoma of the lung, and vinyl chloride, which is associated with the hepatocellular carcinoma exposure. That's all from me. Thank you.